Hey guys, welcome to today's video. It is on inheritance, and this is the last video within Unit 4, Topic 1 on genetics. We move on to evolution and the continuity of life in Unit 4, Topic 2 after this. So here's the learning goals from QCAA. Notice the cognitive verbs here. Firstly, we will predict frequencies of genotypes and phenotypes using data probability models, that being histograms and Punnet squares, and we will also talk about alleles, autosomal dominant, sex-linked, and multiple alleles. We'll define the term polygenic inheritance and again predict frequencies when multiple alleles are involved. So the cognitive verbs here are important to pay attention to. This is what you will be expected to do when you see these terms in the final exam. So to predict means to use the information that you are already provided with to provide a possible hypothesis, an expected outcome. To define is generally 1 to 2 sentences elaborating on or conveying a particular idea or topic or concept. So you can pause the video here and write maybe a summarized version of these learning goals into your books. Okay, let's get into it. So from 11.1 of the text, predicting genotypes and phenotypes. You'll see down the side here, there's a whole bunch of key terms, and hopefully some of these are familiar to you, such as allele, homozygous, heterozygous, but there should also be a number of new terms. So however you are taking notes, whether that's a glossary, pause the video here and get those definitions down. Okay, here's a picture of a man by the name of Gregor Mendel, and if you've ever studied genetics before, you've surely heard that name, probably in conjunction with the term Punnet squares. So Gregor Mendel is known as the father of genetics because of his work with the common garden pea in his monastery. He was a monk as well as a biologist who conducted over 28,000 experiments, and in them he noticed how traits expressed in one generation disappeared in the next, but then reappeared in the following generation, indicating that there was something going on. Genes or material was being passed from one generation to the next. This was a big deal because prior to this, people believed that organisms were simply a blending of their parents' characteristics. So they would have expected, if we're talking flowers, purple flowers to produce purple flowers, or pink flowers to result from the cross of purple and white together. For each of Mendel's crosses, he looked at peas and snapdragons. He used true breeding for the traits he was choosing to study. And a true breed just means if he was using purple flowers as a parent, he knew that was a purple flower because he had crossed purple with purple with purple with purple again and again until he knew, yeah, that's a true breed. I'll introduce you, now we're talking about crosses, to these two terms, monohybrid and dihybrid. So mono, as the name suggests, mono means one. So a monohybrid cross involves only one character or pair of contrasting traits that are studied. So for example, just color. So we could be looking at purple flowers crossing with white flowers, but either way, we're just looking at the one thing, that's color. Whereas a dihybrid cross involves pairs of contrasting traits for two different characteristics. So we could be looking at color and shape. In his monohybrid crosses, Mendel discovered that traits were inherited in very specific ratios. When studying the character of flower color, Mendel truebred parents to have purely white flowers or purely purple flowers. When he was studying these, at first he used the generation that he called the parent generation, where he crossed a purple with a white to produce the next generation of flowers, which is called F1, and F, even though this is the first generation, stands for filial or filial. What was interesting in this monohybrid cross, though, was when he took the offspring of this generation and crossed it with itself, he saw the white that had disappeared in this generation re-emerge in a specific ratio every single time. And that was 1 white flower to every 3 purple flowers. Every time he conducted the experiment, the F2 generation had a 3:1 ratio. And not only had he observed that the white flower trait reappeared, but he discovered that it happened every single time in that ratio. And from his work, he determined 4 main things. Number 1, each individual has 2 copies of a gene for each character, that being each from one parent. And the law of segregation says that when gametes, so sex cells, are formed, they will only receive 1 allele, that's a variant of a gene, for each trait, 1 from each parent. The second thing was there are alternate versions of genes, and today we call these versions alleles. For example, if we're just talking about the gene for flower color, well, it could be either purple or it could be white. Those are the alleles. Number 3, if 2 different alleles for a character occur together, one may have an observable effect while the other may be hidden, and the observable trait is what we call dominant, whereas the hidden trait is recessive, and this is called the law of dominance. Number 4, when gametes are formed, the alleles of each gene separate independently of one another. Thus, gametes carry only 1 allele for that gene. If you've ever studied genetics before, you've probably heard of the Punnet square. Punnet squares are used to predict the probability that offspring will inherit certain traits based on their parents' genotypes. Now, Gregor Mendel did not create or even use Punnet squares when he was completing his work. The diagram used to predict inheritance was created by another biologist called Reginald C. Punnet, who repeated Mendel's work much later in the early 1900s. So if we have a look at the Punnet square now, here we have a couple of questions. This is taken from Khan Academy. Here's the context to the question. Firstly, in dogs, black coat color, represented by uppercase B, uppercase represents the dominant trait, is dominant to yellow coat color. So the recessive trait is represented by lowercase b in this case, and straight fur, uppercase C, is dominant to curly fur, lowercase c. The coat color gene and the fur texture gene are on different chromosomes, so they're not linked. They are sorted independently and are also not sex-linked. Here's the question. In a cross between 2 big B, little b, big C, little c parents, predict the fraction of offspring with the same, we could call that heterozygous genotype. So here's the question in another format. We have 2 heterozygous parents, heterozygous for both coat color and for fur texture. What is the probability of their offspring being of the same genotype? So to answer this question, you can do it 2 ways using Punnet squares. From your readings, you would know that you can do one big Punnet square where you have each allele provided. Generally, the male is up the top and the female down the side. Or you could use the product rule and do a Punnet square for just the coat color and a Punnet square for just the fur texture, and then multiply those together. So either way, you should get the same answer. So if you pause the video here, give that a shot and we'll go through it together in a sec. Okay, I hope that you've had enough time and you have something that looks like this. So the solution. Probability of it being heterozygous. Well, if we just do the fur color first, each parent will contribute a uppercase b or lowercase b, uppercase b, lowercase b. So if we put those together, we have two uppercase b's. That would be one homozygous dominant for fur color. Then we'll have two heterozygous for fur color, and then we'll have one homozygous recessive for fur color. And in that case, it would result in yellow coat color. Similar to fur texture, each would provide their own allele, uppercase, lowercase. Independent assortment says it doesn't matter which one gets which, it's split randomly. So we have, again, a similar outcome where there are two heterozygous outcomes. So if we circle those, we have half multiplied by half. So the chance is the probability of it being heterozygous for both is 1 out of 4. Here's another question for your practice. So in a cross between, again, heterozygous parents, predict the fraction of offspring with black coat color and straight fur. So instead of it asking you what's the genotype, it's asking you what's the phenotype of this. I'll move that over here for you. So pause the video here and give that one a go. Okay, so hopefully you realize that you didn't need to draw any more Punnet squares up because we've done it already, so we can just use that. So this time we're going to look at black coat color. We can see, well, 3 out of 4. And we're going to look for straight fur, which again is 3 out of 4. So 3 by 3 is 9, 4 by 4 is 16. The probability of this occurring is 9 out of 16. That is all for the Mendelian genetics, and we'll move on now to the next slide. So, histograms. Unlike Mendel's pea plants, humans don't come in two clear-cut, tall and short varieties if we're talking about height. In fact, for height, we don't even come in 4, 6 or 8 types. It's this continuous progression of options or data. So that's called continuous variation. It's a variety of phenotypes as a result of more than one gene contributing to a characteristic. So to represent continuous data, we use what are called histograms. And histograms are a bar chart representing frequency distribution, whereby heights of the bars represent observed frequencies. It's a type of diagram that illustrates a frequency of distribution within these rectangular areas. Human features like height, eye color, and hair color come in lots of slightly different forms because they are controlled by many genes, each of which contributes some amount to the overall phenotype. For example, there are 2 major eye color genes, but at least 14 other genes that play roles in determining a person's exact eye color. So it gets a lot more complicated when we're looking at human characteristics generally. Which leads me to the next topic of this video, which is polygenic inheritance. So this is again another non-Mendelian genetic topic. And a definition of polygenic inheritance, as the QCAA learning goal asks for, is this. It refers to a single characteristic that is controlled by more than 2 genes. Examples include skin color, height, and blood type. They're determined by multiple alleles. So looking at a real life example of a human polygenic trait would get complicated, largely because we would have to keep track of tens or even possibly hundreds of different allele pairs, like the 400 involved in height. However, we can use an example involving wheat kernels to see how 3 genes whose alleles add up to influence the same trait, and how this can produce a spectrum of phenotypes, whereby one end of the spectrum is these deep kind of red kernels, and the other end is the white kernels. In this example, there are 3 genes that make reddish pigment in wheat kernels, which we can call A, B, and C. Each comes in 2 alleles, one of which makes pigment, that's the capital letter allele, and one of which does not, that's the lowercase allele. These alleles have additive effects. For example, the lowercase a means that there is no pigment. The uppercase lowercase a means there is some pigment being expressed, and the uppercase AA genotype would contribute again more pigment. And that is the same for B and C genes and their 2 versions of the allele. Now let's imagine that 2 plants, heterozygous for all 3 genes, as we can see up here, the 2 parents, were crossed with one another. Each of the parent plants would have 3 alleles that made pigment, leading to pinkish kernels. So these ones are pink already. Their offspring, however, would fall into 7 color groups, and we can see that kind of spectrum down the bottom here. Ranging from no pigment whatsoever, that's this homozygous, fully recessive state down the bottom, white kernels, to lots of pigment up the very top here, homozygous dominant, where they would be dark red. And this very thing is what researchers have been seeing when crossing certain varieties of wheat. So, takeaway message is we're looking at multiple alleles. So how more than one gene can result in one trait. The takeaway point here is multiple alleles increase the number of possible phenotypes. And we can see that here in this example as well, with fur color of these rabbits from dark gray to albino. So let's look a little more at non-Mendelian genetics that does not fall nicely into what Mendel observed in peas. So here, Mendel's results were groundbreaking, partly because they contradicted the then popular idea that parents' traits were permanently blended in their offspring. In some cases, however, the phenotype of a heterozygous organism can actually blend between the phenotypes of its homozygous parents. This is called incomplete dominance. It has a few other names, intermediate dominance or partial dominance, but we see this in cattle, in horses, and also in different plant species. So here, if we have a chestnut horse that breeds with a white horse, it produces this blend, what we call roan. The other non-Mendelian genetic topic that I want to talk about is codominance. And again, we see it in cattle, horses, but also in plant species, whereby both traits are expressed, not blended, but both are uniquely expressed. So if we have a white camellia and a red camellia, we can see codominance here, whereby there is clearly white and clearly red. These non-Mendelian traits are examples of genetics when we're talking autosomally, if that's the word. It's in reference to the autosomes, so the body cells. The other side of that is sex chromosomes and sex-linked inheritance. So now we move on to 11.3 of the textbook. And if a trait shows up in a family more often in males than it does in females, it is likely to be sex-linked. Since the male only inherits one X chromosome, a sex-linked recessive trait will show up in his phenotype if he has one affected X chromosome. Since the male only inherits one X chromosome. If a female inherits one affected X chromosome, she still has that second from the other parent that is likely unaffected, and that can mask whatever condition it may be. In a pedigree, only females can be carriers of sex-linked recessive traits. I'll show you what a pedigree is in a sec. It's also the same thing as a family tree. So if any of a female's sons are affected, then we know that the mother was a carrier. So here is an example of a pedigree. This is a real-life example of Queen Victoria and a condition that she was likely a carrier of but was unaffected by. But we saw her sons affected by it, so we know that hemophilia is sex-linked. And it's this condition that affects the body's ability to clot wounds. The clotting process, or cascade, is this series of chemical reactions that takes place in the body to like a mesh create this blockade whereby the person doesn't bleed out. And it's a pretty serious condition because people can die just from small wounds. There is treatment for it now though, which is really good. But geneticists use pedigrees, these family trees, to show a family history of an inherited trait in order to help determine the chances of a trait being passed on to the next generation. Each pedigree can show whether someone expresses the phenotype or not. And depending on the parents and all the offspring, you are able to determine the genotype of individuals by setting up Punnet squares. So Punnet squares can work in with pedigrees, and we'll probably do a question on that in class. So here is the probably most classic example of hemophilia. We have Queen Victoria as the carrier. So this was back in the early 1800s, but neither Queen Victoria or Prince Albert had this disease, indicating that she was a carrier. It is also hypothesized that a mutation for the condition arose within her, as none of her predecessors exhibited the disease. But here we can see a few female carriers, and an indication that, again, it's sex-linked is that the males are the ones who are hemophiliacs. They are the ones affected. Just to break down what a family tree is all about, it shows us various generations. The females are represented by circles and males by squares. Whenever you're given a question, you will likely be given a key. In this case, this purple represents hemophiliacs. We can see that from this line here, Queen Victoria and Prince Albert had a number of children. And this daughter, for example, Victoria, had children with Frederick III of Germany. And then their children even had their own children. So that's how we get these various generations. Sometimes you'll see lines through the squares and the circles indicating that that person is now deceased. But we'll talk about a question on this in class. For now, let's get into some questions. So shout out to Mrs. Bagnall from Victory College in Gympie for sharing your PowerPoint with me and these kind of questions. So a little bit different to our normal way of asking questions. So I'll just move myself down here so you can see this. So I'd like you guys to pause the video here to follow these cognitive verbs, to do research if you need to, use the internet or your textbooks, to answer these questions. So keep in mind, when you see define, 1 to 2 sentences, describe, maybe 3 to 4, discuss something similar, 4 sentences, considering an argument, that's the fors and against. Predict is talking about the hypothesized expected outcome based on the data that you have, and explain, you're looking at about 5 to 8 sentences worth of key terms. Once you guys have finished that, and if you're feeling up to it, I know you might have a bit on at the moment, but there's extension. and that is to complete the end of chapter questions once you've finished reading chapter 11. I hope that this has been helpful, and I will leave you with this final slide. Here we have some heterozygotes and genetics. This is how it works. Guys, God bless you. I will see you in class. Bye.